normally for multiple choice you get 1 minute per question, but these are like things you should be able to do, and they're not like application or really thinking, right? You should know them, right? So probably not, right? I mean, like for most of those, you should have read it. It's a sentence and been able to pick the answer, right? That should take you 15 seconds. So then you get 30 seconds. So this is practice for the exams. You're not going to have lots and lots of time to guess and play process of elimination and all. You either know it or you don't. Okay, so the quizzes that have more thinking on them, you'll get more time or less questions. But these were like, you know, what is this phase? What is this phase? That was all the stuff you were supposed to know cold for today. You don't want to wait till next Sunday to study last week's stuff. Right? So if you know that cold already today, then you're just reviewing it for the rest of the week, and those are easy questions on the exam. You're only worrying about Thursday and Friday, the extra stuff that comes. Right? So, yeah. Yeah. Yeah, so no. The answer is no. Any other questions? So yeah, if it seemed too hard or you didn't have time, you need to study more. Okay, because you need to know all this stuff cold. This is the easy memorized stuff, right? You've got to get those points, because the next couple sections are going to be stuff you have to know lots of terms and lots of mechanisms and lots of applications, right? So you don't want to be worrying about this stuff before the next exam, okay? All right, so today, hang on. Today we're going to do meiosis, okay? And we're going to still compare this back to mitosis so you can see the compare and contrast, because you know how instructors love writing compare and contrast questions and multiple choice, right? What are all the things that happen in mitosis that don't happen in meiosis, or what are the commonalities between mitosis and meiosis? Which is the one thing that happens in meiosis that doesn't happen in mitosis? That kind of stuff, okay? So overall, right, meiosis's point is to go from diploid to haploid cells. Right? So if you're making gametes, you're making eggs or sperm, you want them haploid. You want one set of chromosomes. That's what haploid means, right? N equals whatever is the one set of chromosomes. So for us, one number 1, one number 2, one number 3, one number 4, blah, 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 to 22, 1X if it's an egg. And then the same thing for sperm, but either an X or a Y. Right? So half of all sperm have an X and the other 22. And the other half of the sperm have a Y and 22 other chromosomes, right? The normal autosomes. Why do all eggs just have 1 through 22 and an X? That's all we got. Right? I can't make a Y appear in my karyotype, right? Women have 2 X's. We have 2 number 1s, 2 number 2s, 2 X's. There is no Y, right? Things just don't appear out of nowhere, right? Spontaneous generation. So when you're answering these kind of questions, people fall for that all the time. Or they think sperm are all Y. And if sperm were all Y, all there'd be is boys, right? So, again, the memorizing problem instead of the thinking. You know boys have an X and a Y. So their gametes, there has to be X sperm, half and half. When we get through meiosis, you know, you'll see blah, blah, blah at the end. The X goes one way, the Y goes the other, randomly. Half have a Y, half have an X. But women only have 2 X's. 1 X goes one way, 1 X goes the other way. All the eggs have 1 through 22 and an X. So it's the sperm that determines the sex of the offspring. Right? So it's the man's fault if you have 7 boys or 7 girls or whatever that crazy English king that kept chopping his wives' heads off because they kept having girls. Right? That was totally his fault. He was shooting X's. So, you know, but they didn't know. They didn't even know about chromosomes, for Christ's sake. Right? Okay. So So again, the thinking part rather than the, I don't know what's in the gametes, oh my God, boys or why? Right? Just think through the process and you're going to get to the answer. Okay. So meiosis, the big picture. So what do you think, what is number one? What phase that we've already talked about do you think this happens in? Yeah, interphase, right? This is the same thing as before mitosis. Exactly the same thing, except only in specific cells residing in your gonads. Everybody think about your gonads for a minute. Think about the person in the gonads next to you for a minute. Okay, specific cells, spermatogonia, cells in boys, and oogonia, cells in girls. Those are the only ones that can go through meiosis. But those cells themselves are not gametes. They are a normal body cell. They just happen to reside in fun places. Okay? So they are diploid. Right? Every somatic cell, every body cell is diploid, including the spermatogonia, including the oogonia. So we all start as 2N. So meiosis begins as 2N, exactly like mitosis, right? No different. Mitosis starts with a cell that's 2N. Meiosis starts with a cell that's 2N. So don't fall for that. That people get a little crazy and forget that the cell that begins is just a normal body cell. Normal. It's different because it can go through meiosis. The rest of them can't. But it starts out looking genetically like a normal body cell. Okay? So it starts as 2N. G1 is 2N. S phase synthesis. We double the chromosomes. They stay connected, right? The sister chromatids. For us, we'd still have 46 centromeres after replication, right? You start with 46, right? G2, they're the X's. There's still 46 of them. Okay. That's the end of interphase. So interphase is exactly the same whether we're asking about mitosis or meiosis. It's meiosis 1 that's totally different. Okay? So essentially, meiosis itself consists of interphase, meiosis 1, and meiosis 2. So that's why they have them listed as these three big steps. Okay? And so this gives you sort of the main ideas of what happens. Meiosis 1, we're going to learn about the phases, and meiosis 2, we're going to learn about the phases. But they're very similar to the phases of mitosis, so it's nice. We'll talk about how they're different, but the same overall ideas. So the big deal about meiosis 1 that's totally different than mitosis is that the homologous chromosomes, which are the two number 1s, the two number 2s, the two number 3s, the two number 4s, all of those guys, the X and the Y if you're a boy, and the X and the X if you're a girl, those are the homologous chromosomes. They have to find each other, and that's how they end up lining up at metaphase. In mitosis, this doesn't happen. Right? All 46 of ours during mitosis, you can think about lining up in a long line. This number 1 doesn't give a crap where the other number 1 is. Right? One of your number 1s could be down here, the other one could be up here. They don't give a shit about each other. In meiosis, they find each other first in prophase. In prophase 1 of meiosis. And then they line up together in metaphase. The point of that is we want one number 1 and its sister to go one way, and one number 1 and its sister to go the other way. The 2s need to go. So we're separating by sets in meiosis 1. So at the end of meiosis 1, this cell has a full set of chromosomes, 1, 2, 3, 4, 5, all right, to 22, and an X. The other one has 1, 2, 3, blah, 22, and an X, if you're a girl, or a Y, if you're a boy. And so at the end of meiosis meiosis 1, we are already haploid. Again, counting the stupid centromeres. We're going to draw this out together, okay, but just as the overview. And so, remember we talked about to figure out are you diploid or haploid, count the stupid centromeres. So for us, at G2, we'd have 46 centromeres. If we take the 1s and separate, and the 2s and the 3s and so forth, and then we count, we have 23 centromeres after meiosis 1. And we have 2 separate cells. So we go through the stupid phases, and we do a cytokinesis. We have 2 separate cells that are each haploid after meiosis 1. They're still the X's. They have the sisters attached, because we haven't dealt with the sisters yet. But they're haploid because the number of centromeres. Okay. And then we can't stop there, because those stupid chromosomes have their sisters. And if we stopped and let everything dissolve back to spaghetti and those centromeres let go, suddenly they have 46 chromosomes again, but the wrong ones. Not, 2 number 1s, but the same clones of each other. Right? And that's just messed up. We don't want that. So, and they don't do that. They stay together. Meiosis 1's job, Meiosis 1's job is to pull apart the homologous chromosomes. That would be the big thing you need to remember. And then meiosis 2, we need to then pull apart the stupid sisters. Go through all the dumb phases, essentially exactly like mitosis now. Right? We line up, we condense down, we line up, we pull apart, we get to the end, we get our nucleus back, we do a cytokinesis. At the end of meiosis 2, we have 4 cells. The end, that is interesting. Of meiosis 1, we have 2 cells. Yes, sir? The homologous chromosomes. Number 1 from number 1. Number 2 from number 2. Number 3 from number 3. But because we had to do that stupid interphase problem, we had already replicated an S in interphase, so they each have a sister. So we can't just stop at meiosis 1, because we still have too much DNA content. So then we have to go through meiosis 2. Right? Like I said before, it would be so much easier if we didn't do stupid interphase, right? And we just took each stick and went, boink, done. Right? That'd be easy. But that's not how evolution worked. Right? Interphase is exactly the same. We've replicated the stupid chromosomes. So the homologous chromosomes are X's. We pull them apart, 1 and 1, 2 and 2, blah, 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 blah. And then we still have to deal with the sisters in meiosis 2. So if you're going to memorize something, which, again, memorizing is okay as long as you incorporate that with thinking, interphase you always replicate. Meiosis 1, homologous chromosomes come apart. Meiosis 2, sisters come apart. Right? Meiosis 1, you end up with 2 cells. Meiosis 2, you end up with 4 cells at the end. So let's look at their little pictures first from the book, just for fun, okay? And so if they're showing us this guy, so this guy here, they're showing us in G1, okay? And remember we said they have to be 2N, so 2N equals what for this cell if it's a cell in G1? And they're showing them as sausages, but they're just our sticks. It's really chromatin, it's really spaghetti, but they're showing it so that we can count them. Yeah, so this is just 2N equals 2, right? There's 2 chromosomes. And they're showing us different colors, trying to make us think back because we're thinking about heredity, right? When they call something the maternal chromosome or the paternal chromosome, it's where those originally came from in the egg or the sperm that made that organism. So this is a ridiculously simple organism with only 2 chromosomes, right? Just trying to make this simple for you guys, right? So for us, it's 46 chromosomes. 23 of them came from the sperm that made you. 23 of them came from the egg that made you, right? So of your chromosomes, if we looked at the 46, We could paint 23 of them red, they came from mom, 23 of them blue, they came from dad. Right? The only one we really know for sure, if you're a boy, is the Y. You had to get that from your dad because mom doesn't have one. Right? The rest of them you can't tell unless we did some sequencing and looked for like blood type or specific things that are different between them. But otherwise, just looking at them, they don't look any different. They look exactly the same. We're painting them different colors just to keep in mind how this cell was made up. Okay? And it will matter in a minute, just a second. Okay. And then replication, right? That'd be S. And then this guy would be in G2. Right? And so if I were drawing these, I would do this and that. All right. That's our sticks and our lines. And if we're thinking about, we'll put the little centromeres on there, right? Counting our centromeres so we can keep track of how many are there, right? Still 2n equals 2. Still 2n equals 2. So for us, we could think about, okay, they're only showing us chromosome number 1 and the other chromosome number 1. We know there's, 22 more sets in the cell if this were a human cell, but we're just following one set to make it one pair, one homologous pair. Okay? So that's what they're saying here. These two guys together are a homologous pair, number 1 and number 1. The replicated parts are still sisters, just like we named them in mitosis. The chromatids, the exact copies that are stuck together at that centromere are the sister chromatids. Okay, before we do that, let's do this. Now it's down here. Okay, so So if I drew this guy and it was in G1, and we'll just give them their little zits, and I just made them different sizes so we can follow them, right? What would 2n equal? You guys are the best. Okay. And again, red will pretend our maternal, and blue our paternal. Okay? For that, way back when. And then replication happens, and then we're in G2, right? We're still 2n equals 4, because if we count our centromeres, we're good. Okay? And so we could do this for all of the chromosomes, right? This is chromosome 1, the pair. We call these guys homologous. This pair is chromosome 2. Those are homologous to each other. Number 3 and number 3 are homologous. Number 4 and number 4 are homologous. X and Y are homologous. Even though they're really very different, there's enough of the Y that's exactly the same as the X, or a few genes on the Y that are shared by the X, that they can find each other to pair up. If the Y really had all different genes, right, the SRY gene that causes maleness does not exist on the X, so that one doesn't help find the X, but there's enough that are on both, right, that those guys will pair up during meiosis. So those are homologous chromosomes. Okay? If we talk about this guy to this guy, we'd say they're not or non-homologous. Because then, all right, Gus like the mule or the donkey. Gus the talking mule. Anyone remember him? Okay, it was a TV show like a billion years ago. So bad. Okay, right, because it's 1, number 1, and a 2. 1 and 2 are not homologous. So we'd call those the non-homologous chromosomes. They don't give a shit about each other, right? The homologous chromosomes love each other, want to get together. Okay? And then, of course, don't forget, no matter which chromosome we're talking about, the 2 parts that are attached by the centromere are always the sister chromatids. Okay? All right. Okay. So now, if we're here, and I just showed you this, and I told you this was a 2n equals 2 organism, you could tell me what stage of the cell cycle that guy's in. Right? It's 2n equals 2, so you know it has to have 2 chromosomes. Right? And it does. But they each have a sister. So is this G1 or G2? Yeah, so this has to be G2. So they're just ignoring G1. Right? There's G1. Right? Just to keep track. And it had to happen. They're just trying to make their figure simpler, right? Blah, blah, blah. So we had to go G1S and G2. And now they're showing us what happens during meiosis 1 and then what happens during meiosis 2. The overview, skipping the phases. Okay? And so the homologous chromosomes separate. In this case, since there's only 2, there's only 1 homologous pair. So 1 goes one way, so mom, maternal, went that way, and paternal went that way. It's completely random which way the chromosomes go. It depends on which spindle they got hooked up to. And when we do the phases, we'll talk about, right? Prophase, just like in mitosis, is when the spindle starts to attach, puts them to the center for metaphase, right? Just as easily, this one could have went this way and this one could have went that way. It just depends on which spindle grabs them. So for us, with all 46 of ours, right, 23 will be red and 23 will be blue, all the reds don't go one way and all the blues go the other. They could randomly, that's a possibility. 21 reds one way, 2 reds the other, every other one, any possible random combination happens during meiosis, right? We're always talking about one single cell, but again, if you think about your gonads, especially the boys, because that's easier, because sperm, we're just pumping out sperm left and right, right? Girls are kind of just sitting around, so we're not going to think about that. There's lots and lots of cells doing meiosis. One individual spermatogonia is picking, right, 10 reds and, whatever combo of reds and blues. The one next to it is doing something different. Maybe all the blues went one way and the next one next to it, 3/4 red went one way, 1/4 blue went the other. So each cell is doing its own random thing about which number 1 went one way, which number 1, the number 2s went that way, the number 3s went that way, the number 4s went that way. Right? So every single cell going through meiosis is randomly doing a different set. And so if the red was the maternal, so that'd be your mom, which would be your offspring's grandma, right? Right? Think about that. And then the blue one in you came from your dad, which means if you have a baby, that's grandpa's, right? So baby might get grandma's nose and grandpa's ears, right, which came through you. You may have grandpa's nose, but you still have the allele for grandma's. It just didn't show up, and so grandma could show up in your kid even though it didn't show up in you, right? So great-great-great-Aunt Edna, who had a really ugly chin, could end up on your poor child, right, even though it's hidden for generations and generations and generations. Okay. And so then every egg you're making, every sperm you're making, has a different combination of all these different chromosomes, right? And only half of you, right, each sperm only has half of you, and which half you got how many reds, how many blues, is random. So theoretically, if all the blues did go to that sperm, that kid's going to look a lot like grandpa, or act like grandpa, or have metabolic disorders like grandpa, or whatever. Right? Randomly more than a sibling that may get more reds than blues. Okay? So much fun. I think about great-grandma Edna's chin. Okay. So both of my children were ginormous at birth. And they're still above average for height. And my husband's family is a bunch of shrimps. I had said he's going to run to my gene pool. But nobody else in my family, my dad was like 5'11", and, I used to be 5'8", I've shrunk over the years. But not huge people, but my kids are like on the trajectory to be enormous people, right? And so, doing a little genealogy, my great-grandmother, so their great-great-grandmother was 6 feet tall. And her brothers, because they all had like 14 children or some crazy, they're farmers. Wow, man, those are tough people. Holy crap. We're all 6'4", 6'5", whatever. So those genes somehow dumped into both of my kids, right? The odds of both of them getting that is pretty rare, considering chromosomes going randomly, blah, 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 and only having 2 kids, right? If I had 10 kids, you think maybe one would pop out like that. But none of my other cousins, and there's lots of them, had, I called it giant baby syndrome, right? Where we had to kick them out early because they were ridiculous, right? And it's not good. So, right, things that are hidden way back out there can show up, right? Good things, bad things, so it's all fun. It was ridiculous. I was enormous. It was disgusting. Oh my God. No one can see those pictures. It's so gross. It's just gross. Yeah. Really bad. Not good. Drugs are good. Drugs are very, very good. Okay. We'll have more of that later. Okay. So that's essentially Meiosis 1, right? The grandma and grandpa, which way are you going to go? Okay. And then Meiosis 2, we're just finishing the job so that we don't have too much DNA content, because we did that stupid replication up there in Interphase that we couldn't avoid, right? And so here, we're already at n equals 1, right? If we count our centromeres, there's 1 there and 1 there. So the first cell division in Meiosis 1, is where the reduction, they call it the reduction division, because we went from 2n to n, right? 1 set of chromosomes, 1 number 1, 1 number 2, 1 number 3, 1 number 4, blah, 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 1 y, or 1x. And then from here to here, we're just going n equals 1 to n equals 1, which seems stupid, and that does make people's heads get a little spinny, right? Because we're taking this guy and we're essentially doing mitosis. Spindle grabs the kinetochore on either side, one sister goes one way, one sister goes the other way. Okay? And so then, if we count just centromeres, each one of these still only has 1 centromere, so we went n equals 1 to n equals 1. So the reduction division where you go 2n to n is always Meiosis 1. That make sense? And again, so just like, draw your stupid centromeres and count those as far as knowing how many chromosomes. Okay? Okay. Ooh. Let's do some drawing. And then we're going to screenshot and upload to whatever's in today. I don't even know. Okay. So let's start, with a cell in G1, and let's give it 2n equals 6. We haven't done that, guys. Oh, I'm going to run out of room. And then we want to see G2, right? And then we want to see Meiosis 1, and then we want to see the end products of Meiosis 1, the end products of Meiosis 2. And we want, as usual, we want the unreplicated as a line, the replicated as an X. And we want those right? The correct number and the correct shape in each one of our cells. And then you can also add your ploidy. So we know this starts at 2n equals 6. So give me, what's it for this guy, and that guy too. And then we'll screenshot and upload. You don't have to draw the chromosomes looking any different or in different colors, but if that helps you thinking through it, you can, and I'll try to remember to do that for you when I go through it. On the exam, it doesn't, it's just going to be a pencil, right? You're not going to bring a bunch of colored pencils or highlighters because you don't want to spend 7 hours just drawing pretty chromosomes. But you can, you don't have to here, but you can if that helps you keep track of which ones are the homologous ones, who gets together, that kind of stuff. And if you're stuck, you can wave me down. I can give you hints. Yep. Yep. So the other thing here's another good idea. So this looks, I was going to say exactly, similar to what shows up on the exam. And so even if you panic, you have an attack during the exam and you cannot think and every piece of information has left your head, except some of the memorized stuff. So that can help you. So you can say, okay, the point of meiosis is to go from 2n to n. We start with one cell, we end with 4. So you can start one cell at the top and 4 at the bottom. Right? That's how you could start your question and then fill in, because you're going to get points for what your cells look like and the correct number of chromosomes and their shape. So even if you don't do the middle, you can still get credit for the beginning and the end. So if you're 2n equals 6, what are our gametes going to be at the end? Yeah, n equals 3. So you could start with that, right? So if 2n equals 6 in G1, we know that's just 6 sticks. And at the end, if n equals 3, we know that's just 3 sticks. So you could start there, and especially if you're panicking, that will help you remember, okay, now I remember, how do I get to 3 if I start with 6? I know G2 has to be X's. Oh, now I'm remembering meiosis 1 is the homologous chromosomes come apart. Well, in order to go from that then to the 6 at the end, the sisters have to come apart. So that can help you think through the problem, right? Because 2n is 12, n has to be 6. 2n is 10, n has to be 5, right? You can just do that. That's what we memorized, right? All the stupid 2n to n stuff. What do you start with? What do you end with? Then think through the steps, right? You don't have a blank piece of paper in front of you. That also helps reduce panic. Don't panic. Okay, so, right, so from here to meiosis 1, there's only 2 cells. Then we get to the 4 at the bottom, right? We have to go through meiosis 1 and meiosis 2.
So this isn't right.
So this is okay, yep. 2n equals 6, yep. And then from this stage, there's only 2 cells. Then there's 4 cells.
Okay.
Yeah.
Because I drew a bunch of different bubbles. Wait
a minute. Yeah. So there's 2 here, and then there's 4. So meiosis 1, there's 2 cells. Meiosis 2, there's 4.
Okay. Thank you.
Yeah. Thumbs up.
Yeah.
Yep. Bueno. You guys are awesome. Yep. So this one, right, this one we have to stay with, so this is G2, so this should be meiosis 1, so the X's, these should still be X's. Because the homologous chromosomes are going to come apart.
Should they all be right and all be
You could do, technically they could be whatever, they could be a whole different combination. Right, it just depends on which way they go. They could be all blue and all red, they could be 2 blues and, yeah, they could be 2 blues, 1 red, they could be 3, that's the randomness. For this, you can do whatever you want, but just in general, yeah, so then those are the X's, right, and then you separate the sisters into the 4 cells. Yeah. Get all the way to the end at the very end. Yep. And then you pull apart the sisters for each one. Right, so the sisters come apart to this one. So then
there's going to be 3 6 in each, right?
there'll be 3 in each, right? Because you wanted to go from 6 to 3.
Oh.
Right, so that's like, so 2n equals 6 at the beginning, you want to end with n equals 3. So if you write that at the bottom, n equals 3, what you know, if I just tell you draw a cell that's n equals 3, it's just 3, 6. And then you can work through the, work it, people, work it. And then screenshot and upload, and then I'll go through it. So even if you didn't get it, that's awesome. Okay. So I'm not going to correct these online because that's too hard, but I'm going to go through it here, so then if you already screenshotted it and uploaded it, now you could take notes on it and give yourself a thumbs up or go, oh crap, and change it. Yep. Yep. Perfect. Okay, so G1 you have 6 sticks, right? And we'll just put their little bumps on them. And we know they're really chromatin and blah, blah, blah. And in this case, right, the homologous chromosomes don't need to be anywhere near each other. They don't know where each other are, they're just out there. And then each one becomes an X. Oops, that's a Y. Okay, right. So G2, right, this is just exactly the same as mitosis. So 6 centromeres up here, 6 centromeres here. We're still at 2n equals 6. And the end of meiosis 1, we end up with 2 cells. And the end of meiosis 2, we end up with 4 cells. Okay, so again, if you're panicking about the chromosomes, you can still get the number of cells. Right? So for each cytokinesis, it can only split a cell in half. So we can think about meiosis being 1 replication, right, in interphase, and 2 cytokineses. The end of meiosis 1, we cytokinese. The end of meiosis 2, we cytokinese both of those that cytokinesed. Right? Sorry. So we start with 1 cell. We go through and end with 4. Okay. So at this step between G2 and the end of meiosis 1, we pair up homologous chromosomes and then we pull them apart. And so we could take, right, like we said, all the maternal could go one way and all the paternal could go the other way. Right, that's totally fair game. Or, oh, stop it. Or they could separate that way. And now I'm going to be lazy. We're just going to cover that guy like that. We're going to cover this guy. Or they could do that. Right? All those different combos. And so for us, if we have 46 chromosomes, so 23 pairs, that's a lot of combos between those just at that step. Right? Grandma versus grandpa. Okay. And then the next step is just, right, 1 sister comes here from the red guy, 1 sister comes there. 1 from that blue, 1 from this guy, 1 from that guy. That's just mitosis, essentially. Same process. So the blue separates, that red one separates, the other red one separates. If we go back and say, ooh, what's our ploidy? Right, there's our centromeres. What's our story for this guy? Is he haploid or diploid? Yeah. And what's his number? You betcha. And what about these losers down here? Yeah. And they got 3. It's simple. Well, not really. That make sense, though? Okay. So if you guys practice this with all the possible numbers, there's really nothing I can do on the exam that you won't know how to do. Right? The other thing I wanted to do, let's just play that little game really quickly. And let's do a picture. Yes, let's have another upload too, because you know how much we love that. Oh, wait a minute, I did the wrong thing, I'm a loser. G1. And let's go with one of those, let's pretend like we're a mule, because you know how we love mules, except that we are going to not use 63 chromosomes, because that's stupid. We're going to pretend like we're a mule that happens to only have 5 chromosomes. Okay? So our mule, we did the 2n equals 5, and we did mitosis on it, I think, in class Friday. If we didn't, you guys can think through mitosis, right? Which, as it turns out, is exactly like meiosis at the beginning, so let's just do that. So how many sticks do we have in G1? And then, are they 6 or X's in G2? Well, you guys are the best. 2, and there's 5 of them. Yes. No spontaneous generation. We can't add a chromosome. We can't lose one. Right? Conservation of matter. Okay. So if we were mitosing, we would just pull the stupid sisters apart, and we'd have 2 cells, each with 5 sticks, right? And so that works fine. That's why the little mule guy can go from a fertilized egg to a full-grown mule, because it's after fertilization, it's just mitosis, blah, blah, blah. If that poor mule tries to now think about the mule's gonads, everybody conjure up an image, okay? We'll go with spermatogonia of the mule. And so now we'll pretend like it's the mule spermatogonia here. And it's going to try to do meiosis, right? Because it's a cell. It's getting the signal. It's trying to make sperm, okay? So for meiosis 1, right, we have to pair up the homologous chromosomes and then pull to the other side. So if we were going to, we'll go do like ... Hang on, let me erase this. Ooh, hello, I'm back. Right, so we're going to try to pair these guys up. So those 2 want to pair, and these 2 want to pair. Oh, the 5th wheel. That's really sad. Oh, look at him. He doesn't have a partner. So usually, meiosis will stall. Right? It's just like during mitosis, there's checkpoints, right? Because if it tried to do this, it would pull, it would end up, right? It's not going to be even. Right? That's going to be a problem. And even if it didn't stall and it tried to get all the way through, the gametes are all screwed up. Yeah. So if they are fertile, they're getting through meiosis, right? But then look at these gametes, right? This is still 2n equals 5. This one's n equals 3, and this one's n equals 2. Right? And same with these guys. This is 3, and this is 2. So if this is the boy mule, then he also has to find a girl mule that happened to make it through meiosis, right? But if, we'll make this guy the sperm, since I said we were thinking about gonads there, right? If that one with an egg that only had 2, the half that got 2, this thing is going to make a 2n equals 4, which is not a mule, right? It's something else, which won't have enough chromosomes to build that organism, right? If 2 number 3s, right, if this is a sperm and there's an egg that had a 3 gets together, right, that's 2n equals 6. Again, not a mule. What's that going to build? Right? Doesn't have the exact right complement of chromosomes. And then even if a 3 and a 2 ended up getting together, yeah, you end up being 2n equals 5, but which 3 and which 2, did it work exactly the same? Which one got left out in that original organism? Yeah. Yeah, so it's still not all the right chromosomes. So it's within the realm of possibility. If you did IVF and you really worked hard, you could get mule-on-mule action for baby mule. But that's a hell of a lot of work. Just go back to the horse and the donkey and go the old-fashioned way to get another mule. Yeah. So it almost never actually goes. But there are evidence of mules that can make sperm. Right? So obviously, if you make a few sperm, some of those cells got through meiosis. But they're not going to be, n equals 2.5 doesn't exist. Right? So, yeah, and you just say they're sterile. Yeah. Because most of the time it stalls, it's not balanced. There's no place else. The other spindle is trying to still connect to that homolog. It can't do it. It just stalls. Yes, do a screenshot and upload for fun. For extra points. Yes, sir.
With all meiosis, whenever the diploid is an odd number, it's going to be sterile.
Yeah, exactly. So let's think about this. Let's think about plants, our good little friends, the plants. Who can think of some plant that stalls in meiosis? So if you're a plant, we didn't really talk about this yet. If you're a plant, you end up, after fertilization, what do you make? You don't make a baby, right? You make a seed, right? Fertilization in plants, egg and sperm get together, sexual reproduction. Plants can do themselves, right? So they're a little bit fancier than we are. Right? Most plants are both male and female parts. Parts gets together. And then they make a seed. So if you stall during meiosis, if you're an odd polyploid and you stall during meiosis, you won't make seeds. What kind of yummy fruits are out there that don't make seeds? Grapes. Seedless grapes, right? There's lots of grapes that have seeds, but there's seedless grapes to eat. Interesting. Check its chromosomes. What about bananas? Bananas have little tiny, seeds. That was for Stein. She said I'm not cursing enough. Right? What about watermelon? Normal watermelon have big old seeds. You can have a seed spitting contest. Seedless watermelon, they're little loser seeds. So they don't develop correctly because they didn't do fertilization. Right? They still go through some of the parts of building the seed, but it's not, you plant those, nothing's going to happen. It doesn't have both parts. The black ones probably developed a little farther, and so I'm not sure if those, I don't think they're functional because they're way too small, but they may have been fertilized, but because they're not all the right chromosomes, they just kind of, but I'd have to look for sure. Because, there's the white ones, and then occasionally you find a black one. Yeah. And what about navel oranges? But every once in a while you find a pretty good-sized seed in there. That one likely made it through. Okay. So how do we propagate more of those plants? We can't plant their seeds. We have to go back and do the hybridization or do the grafting. Right? Plants are pretty easy. You chop a branch off, you can jam it in the dirt and it'll grow roots. Right? Some plants are easier than graft it to another tree's roots. Right? Grow a new plant. Because plants have stem cells throughout all of their tissue, very similar to embryonic stem cells. We don't. I can't cut your arm off and stick it on somebody else and really make it happen without lots of work. I I can't stick it in the ground, a cup of water, get some body parts off of it, right? Okay, so that's just some fun facts about meiosis. We love meiosis. Okay. Oh my God, I forgot about this. So we were talking about this last time, and I thought this was in this lecture, but it's not, so I'm going to add it just really quickly. We talked about non-disjunction, where the chromosomes, there's the right numbers in there, but they don't separate correctly, okay? And so usually that's a defect in formulating that spindle apparatus and getting the spindle to grab the centromeres or the kinetochore. And you can have a non-disjunction event in either meiosis 1 or meiosis 2. So they're both possible. In one meiotic process, it's unlikely to have a non-disjunction in both 1 and 2. It's within the realm of possibility, but it's pretty rare, so it's unlikely to non-disjoin in 1 and in 2. So I'll give you a little example of that, because that was when we were talking about the XYY. How do you become XYY? How do you become triple X? How do you become, trisomy 21? What the hell's going on? So if we start the cell, another cell, we're going to put them close together. So G1, G2, and we're just going to do 2n equals 6, because then I don't have to draw a million things. And so the end of meiosis 1 and the end of meiosis 2, if there's a non-disjunction event in 1, that means the homologous chromosomes don't all line up correctly, don't connect to the right spindle, and don't pull apart. Usually 1 just doesn't connect. It's not that it connects to the wrong side of the spindle and gets pulled the wrong way. It's usually just a loser that doesn't get attached to anything. So if we were pulling, let me change back to my blues. Right, we're supposed to get 3 of each, but 1 gets left behind. Let's say this guy was the loser, and he just didn't attach to the spindle, and he just happened to be on this side of the cell. So when the cytokinesis between it, that one just happens to end up in that cell. Okay, so it's usually just not attached, and it just ends up with wherever it happens to sit between the pinching off. Remember the cleavage. Don't forget about cleavage, people. Very important. Okay, so now this is screwed up, right? So if this was 2n equals 6 up here, what the hell? What is it supposed to be? N equals 3. Are any of them n equals 3? No, they're all effed up. Right? So then so then we're going to assume meiosis 2 goes correctly, and the sisters come apart, 1 of each. Right? The gametes are a mess. So if this one ends up being a sperm that fertilizes a normal, ooh, look at that. Right, there's a little sperm guy. We'll say this is a normal haploid gamete, supposed to have 3, right? And this sperm gets jiggy, and we get that is not good. What does that 2n equal? Yes. Is that 2n equals 6? So if this were a human being and we're pretending instead of 6 it's 46, and this extra guy is chromosome 21, right? So this guy is chromosome 21, this guy is chromosome 21. Oops, sorry, I'm dumb. This one is chromosome 21, right? That means this is a 21 and this is a 21. And so that's trisomy 21. You got 2 21s from the sperm, 1 21 from the egg like you're supposed to. The organism has 3 chromosome 21s. That offspring has Down syndrome. Okay? So that's a non-disjunction in meiosis 1. If we look at these guys, makes one of these guys a little happy sperm, and he tries to get together with a normal haploid egg, right? And they have the little offspring, right? That's 2n equals 5. That's not 2n equals 6. That would be if we were talking about chromosome 21. Now this one only has one chromosome 21, nothing else. The rest are fine. So you'd have 45 chromosomes. That does not survive. Okay. This one would have 47 chromosomes, right? If it was a human, we were talking chromosome 21, that has Down syndrome. You can't be missing any of the autosomes. You can't be missing a 1, a 2, a 3, a 4, up through 22. You cannot be missing that. Okay. You can be missing an X or a Y. You can be 45X nothing. That's Turner syndrome. Looks pretty phenotypically normal, but not a functional reproductive system. Okay, so that's, yes. Yes, they can happen to any of the chromosomes. We just don't ever see them because they don't result in a pregnancy, right? So we only see trisomy 21, trisomy 13, and trisomy 18 because they survive, or at least some have survived to birth. They're 13 and 18 don't survive more than months to years, and 21 survive, can be fertile, right? Can be. So that's meiosis makes it through sometimes. It depends on the severity. Like if you look at trisomy 21, phenotypically, there's all levels of genetic anomalies. Some very slight, very high functioning, very normal. Some very not. And that's just got to be the combination of alleles on those three and what they're doing. Yeah. Okay. Yes. Yeah, so meiosis 2 non-disjunction can happen also. So I'll do that as fast as I can so we don't waste too much time. 5, 6. 1, 2, 3, 4, 5, 6. Meiosis 1 is normal. Usually, so this was 1, this is the end of 2, G2, G1. Right, 2n equals 6. So in this case, we'll pretend like this side goes normally. Because again, if you have a non-disjunction, it's unlikely that both sides will go bad, just usually one. So this one we'll say is normal, right? That's n equals 3, that's totally fine. But this one, the spindle doesn't attach to both sides of that centromere at the kinetochores, and so one X goes completely together, right? So we'll show it like that, and then the other ones are okay. So you end up with an n equals 4 and an n equals 2. So if either of those two gametes are involved in a fertilization reaction, they're screwed up, right? They're going to either be missing a chromosome or have too many, right? If that was 21, 3 21s coming from that one, or this would be not enough 21s, right? So this would be, if it were 21, that would be a down. This would be dead, and these are all normal. So a non-disjunction in meiosis 1 screws all the gametes. In meiosis 2, it really only screws up 2 of them, half of them. So it's less severe, right? So this could happen, and we'd never know it, because if this is the sperm that wins the swim, it's all good. It's all about the swimmers. Or it could be in the egg, and then if that's the one that happens to pop out the month that the sperm get lucky, right, you'd never know that you had a non-disjunction going on. It's only, you know, this side that went bad. Yeah?
Down syndrome sperm getting normal sperm, I don't get it. It's just like a race. Like, I don't get how.
Is it a race?
Like,
It is a race. Yeah. Well, there's a lot of luck going on though too. Yeah, so theoretically, the best swimmers should get up the canal and all through, but some of it's kind of random. You get kind of yanked in there and sloshed around, and right. So, and then once they're there, they're all digging through the jelly. So even if you got there first, but then you're a great swimmer, your flagella is just, on fire, but your acrosome that digs through the jelly totally sucks, somebody else can beat you into the actual egg. You got there first, but then you farted around, doing nothing, and they all, So usually thousands of sperm make it to the egg. They're all digging. One digs the best first. So you have to swim fast and be a good digger. Survival of the fittest. And then randomly, some loser could just, mooch off the guy next to him and then, whoa, zip right in there. Yeah. Right. Yeah, so this is in Meiosis 1, non-disjunction. Yeah, all of them are screwed up. And then non-disjunction Meiosis 2, half of them are screwed up. So age is a factor because your eggs get old. It can happen in sperm as well, but they're not sure if that's correlated with age or with other health issues in men. They do have evidence that non-disjunction happens in sperm as well. For women, when we get, oh my God, we're not going to get to the end. If you go through the rest of the slides, our eggs actually stall in Meiosis 1. We don't finish and go through, and we actually stall even before Meiosis 1 is complete. Upon ovulation, we finish Meiosis 1, and then upon fertilization, we finish Meiosis 2. So they think, we start with 1 million eggs. At birth, we have 250,000 functional. By the time we're 40, we have 10,000 functional. And if you talk to IVF doctors above 40, 42, only like 1 in 10 are actual eggs that will function and become an embryo. Most of the rest have chromosomal anomalies. So they think it's been sitting around for so long, then all of a sudden, oh my God, it's my turn to finish meiosis, and it just doesn't work right. Okay, so what else? Oh my God. Okay, so this is fertilization, right? Just like we talked about. This goes back to their original example where they had 2n equals 2, right? So this is n equals what? 1, n equals 1. And now, guess what? This little happy guy is back to 2n equals 2. And then in the corner, this is just a picture of the egg with a whole bunch of sperm attached to him. So the egg is enormous, and the sperm is just chromosomes with a capsule, and a tail trying to get there. So the egg provides all of the organelles and all of the proteins and all of the stuff needed to make an offspring happen. But it has to have the sperm DNA to get to that diploid number. Okay, so outcomes of meiosis. 4 daughter cells, right? That's what we just call the stupid, cells. It's just like mitosis, 2 daughter cells with the exact same number of chromosomes as the parent. Meiosis, 4 daughter cells, half the number of chromosomes as the parent. Then they have to do some maturation to become a functional egg or a functional sperm. And then, yay, this is, right, woohoo. Okay, so let's do the phases like quickly, fast, super fast. And then we'll finish the rest of the complicated stuff tomorrow. So interphase, we already talked about, exactly the same. G1, S, G2, boom. Prophase 1, again, just like mitosis, we condense down the chromosomes, right? The nucleus breaks down, so none of this is different. The spindle starts. This is different. Synapse means the homologous chromosomes get together. The homochromos dig each other. Dig it. Okay? Right? They get together. They line up. They pair up. So So this is where they pair up. So they're showing them here, right? So they're saying this guy, right, this is 2n equals 4. This is homologous pair 1, homologous pair 2. Two number 1s, two number 2s get together. So a 1 and a 2 aren't together, right? 1 and 1, 2 and 2. If it's a human, 3 and 3, 4 and 4, 5 and 5, 6 and 6, blah, 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 blah. Okay. So we don't have a pro-metaphase in meiosis, but they went with early prophase 1 and late prophase 1. I don't really care. It's all prophase 1 to me. Crossing over is super important, right? Today, we're just going to remember that it happens in prophase 1. Homologous chromosomes align, pair up, and crossing over happens. We'll talk about what the hell that is tomorrow. Chiasma is just the actual crossing over physical part. You can actually see it under a microscope, right? Because they're fat, chunky sausage chromosomes. You can actually see where they cross. That crossing point they named chiasma. Right? Ooh, yes. These guys start to separate, go to their poles. Okay, we're going to do that tomorrow. Metaphase, line up in the middle. But now they're lined up in pairs, right? So that was the whole point. If you pair up in prophase, you're not going to unpair up and line up. You stay paired, lined up. So the spindle attaches to this centromere to go that way. This one goes to that side. This one goes that way. This one goes that way. And remember how they pair up. The reds could be on the same side. The blues could be on the same side. They could go any old way you want.